Quickly tell me in the chat, students, if I'm audible and visible to all of you students. Quickly let me know in the chat right now if I'm audible and visible to all of you students. Welcome to Vedantu Science Olympiad. This is Baswaraj sir, your biology mass teacher students. Welcome back. Now, today morning we did the entire plant kingdom, and when I was researching multiple previous year questions, plant kingdom questions were comparatively less. So I decided, okay, I believe all of you, har ek bacha na, at least you have finished the first one, right? Once again, students, 1 second. Oops. At least all of you finished the cell biology. Yes or no? All of you students? Yes. All of you finished cell biology at least once in your life. All of you. 1 second, students. 1 second. There is something wrong with the data. All of you would have finished cell biology at least one time in your syllabus right now. If you watched my lecture, any lecture, you might have finished. So let's try to solve some questions from cell biology, which includes your entire cell structure and also mitosis and meiosis in this particular lecture, right, students? Okay. Now, all of you ready, students? All of you ready? Yes? Yes? Ready, all of you? Okay. Now, students, I will finish everything, students. Don't worry. Next, if you do not know what is mitosis, what is meiosis, ask me. If you do not understand mitosis and meiosis, I will try to cover up mitosis in 30 minutes. Meiosis in 30 minutes, I'll make it for you in the coming days. Okay. Now, this is a question, student. This is a question from NSEJS 2022, which we discussed in today's class. In today's class, this concept was discussed today, right? So, what is the question? The question says, right? This is from plant kingdom, right? The Carnelius Linnaeus, a Swedish botanist, is credited with botanical nomenclature. He was given as botanical nomenclature of plants and animals. While using binomials, he devised a system of classification of plants. His classification was, students, do you remember I told you his classification was mainly from where? Mainly from outside face, outside body, flower color, leaf color, leaf shape. But in his case, it was andrecium structure, collection of andrecium. But all of this is what, students? All of this is artificial system of classification. Can anyone in the chat tell me? Can anyone in the chat tell me who gave natural system of classification? Anyone? Try, students. Try. Remember, I told you two scientists. Remember, I told you two different scientists who are involved in the natural system of classification. That was who, students? Bentham and Hooker. Bentham and Hooker are involved in natural system of classification. Please remember that. Yes, Bentham and Hooker. Now, students, next question, all of you on the board, ready? Here is the next question. This question is from NSEJS 2015-16 year, right? The genetic material DNA in plants occurs in which of the following cell organelles? Which of the following cell organelles we can find the DNA material? We can find in nucleus. Yes, we can find in nucleus. We can find in chloroplast. Yes, we can find in chloroplast. We can find in mitochondria. Yes, we can find in mitochondria. Because mitochondria and chloroplast, they have their own DNA. So what is the answer, students? What is the answer? Answer is option number C, obviously. Answer is option number C, right? Next time I'll give you more time, students. I realized I didn't give you time. Next question, I'll give you more time to answer. Okay. Now, here's the next question. Take your time. Students, here's the next question. All of you students, if you're able to solve the question, quickly smash your like button, all of you. Quickly smash your like button, all of you. All of you. Quickly smash your like button if you're able to solve the questions. All of you. This is the next question. I will give you exactly, students. I'll give you exactly 1 minute time. I'll give you exactly 1 minute time. Here we go. Aram solve karo. I'll read the question for you. In a human cell undergoing meiosis, so reduction division, at what are the total number of cellular DNA molecules present during the prophase 1? So during prophase 1, how many DNA molecules are present? It's a very, very good question. It's a very good question. Okay. So I will teach it to you one by one. So ready, all of you? Ready? Answer is not A. Answer is not A. Take your time, students. Take your time, because if you want to understand this, you need to understand meiosis a little bit. I'll teach you also right now. Okay. Take 1.5 minute. All of you can take 1.5 minute. A and B both are wrong. Take 20 more seconds. Take 20 more seconds. All of you. It's a very good question. Such questions can be asked. Such questions can be asked multiple times. So you need to know if there is a variation of it, will you be able to answer? Okay. Okay, students. Let me explain, students. Let me explain you. All of you know meiosis. All of you know meiosis. So this is how I teach generally. So this question would be easy for you if you're my student. Okay. Now this is how I teach this entire chapter at the end. So we know that we have G1, S, G2, prophase, metaphase, anaphase, telophase. All of you know, right? All of you might be aware, students. All of you should be aware that there is something called as G1. There is something called as G1. After that we have S, then we have G2. After the entire initial thing, after that we have the PMAT. P-M-A-T. So students, what is PMAT, all of you? What is P? P stands for prophase, M stands for metaphase, A stands for anaphase, and T stands for telophase. This much all of you know, PMAT. All of you know what is PMAT? Okay, all of you know what is PMAT right now. All of you know what is PMAT. Now I am talking about prophase 1, and it is part of meiosis. Now in meiosis, all of you need to know what? In meiosis, all of you need to know that we have G1, S, G2. After that we have something called as PMAT 111, prophase 1, metaphase 1, anaphase 1, telophase 1. After that we have something called as interkinesis, right? Interkinesis. After that, now again we have PMAT 222. So prophase 1, anaphase 1, telophase 1, prophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. This much all of you need to know. So 111, 222. Now the question is here, prophase 1. Prophase 1, what is the DNA content? How many molecules of DNA are present in prophase 1, they're asking you. Okay. So for that, you need to know this entire table for it. Now let's say in G1, number of chromosomes is 46 chromosomes. We are diploid organisms, 46 chromosomes are present. Every single chromosome has how many chromatid in the starting? Every single chromosome has only one chromatid here. If all of you notice here, students, in the starting, every single chromosome has one chromatid here. So number of chromatids is 46. DNA content is 40. This is just an example. Here. And 2 and 2C is the condition. 2 and 2C is the condition right now. Now in the S phase, what happens? In S phase of your cell cycle, the DNA replication takes place. DNA replicate हो रहा है. कहाँ पर? S phase के अंदर. S phase के अंदर DNA replicate हो रहा है ना? So when DNA is replicating, the number of chromatids are increasing. The DNA content is increasing. Where? In the S phase. But if the DNA content is increasing, if you can notice here, one chromatid became two chromatid here. One chromatid became how many chromatids? Two chromatids right now. Two chromatids. Okay. So number of chromosome will remain the same. Number of chromosome will remain the same. But number of chromatid is becoming 92 right now. 92 number of chromatids. Okay. So we need to understand number of chromosome will remain same right now. Remain same. Will remain same here. Can you see? No change in chromosome number here. At least until telophase. Okay. But number of chromatid is becoming double. हर एक क्रोमोसोम को दो क्रोमैटिड हो गया. So 46 into 2 is how much, students? What is 46 into 2? Anyone? What is 46 into 2? Anyone? Please, students, in the chat. One chromosome has two chromatids. 46 chromosomes will have how much? 46 chromosomes will have how much? 92 chromatids. So अभी totally एक सेल के अंदर अभी 92 क्रोमैटिड्स हैं, क्योंकि हर एक क्रोमोसोम के पास 2 क्रोमैटिड है अभी. So there are 46 chromosomes, 92 chromatids. 92 chromatids are present because every single chromosome has 2 chromatids right now. So the same is maintained in the prophase, metaphase, anaphase. Same is maintained. So students, what is the answer, all of you? What is the answer? The DNA molecules present at the prophase 1 is going to be what? Prophase 1 is going to be what? 92 right now. So students, I'm going to ask you one more time. Do you want a short video on only mitosis, a short video on only meiosis? Because if you want to solve the questions on meiosis, you need to understand the depth of it. You can't just by heart anything. This cannot be learned by hearting. Not possible. You can't by heart any of this. See, biology, everyone believes by hearting, but it is not by hearting. It is understanding the concept of meiosis and mitosis, right? Now, next question is easier. All of you solve this question quickly. Students. It's very interesting. Very interesting, because many students don't understand meiosis. They mug up meiosis. Meiosis is so beautiful. What happens G1S, G2? What happens prophase, metaphase, anaphase, telophase? In prophase 1, what do we have? Leptotene, zygotine, pachytene, diploidine, diakinesis. What happens in leptotene? What happens in zygotine? What happens in diakinesis? That is very important. So you can't by heart all that. You have to visually understand. You have to visually look at it. Okay? Now, which of the following is not produced by the endoplasmic reticulum? Students, all of you should know. ER is of how many types? Students? ER is how many types? All of you. We have RER, rough endoplasmic reticulum, and SER, smooth endoplasmic reticulum. Smooth and rough endoplasmic reticulum we have, right? The rough endoplasmic reticulum mainly produces, it is called as rough. It is called as rough because of ribosomes. Okay, because of ribosomes. Now, it is called as smooth because of no ribosomes here. And here, it can, since ribosomes we have here, it can produce proteins. Proteins can be produced. Here, it mainly produces lipids and hormones. Lipids and hormones are produced from here. But do you see anywhere glucose production? Do you see anywhere carbohydrate production? No, we do not. Carbohydrates are not produced by your ER. So option number C is the right answer. Okay. Next question, student. Next question, very easy question. Student, next question. Let's see who tells the answer first. All of you. Let's see who answers the questions first. I want to see. I'll take the name out. We'll see. We have Ruturaj. We have class 9 to MIT. We have Vivain. Yes. We have Ayush. We have Arsh. Yes. We have Tejas. Students, it's a very easy question again. Very easy question. Which among the following is not a plastid? Now, if you want to understand the number of see, in a cell, we mainly have 3 types of plastid. How many we have, students? 3 types of plastid. One is called as a chloroplast. One is called as a chromoplast. One is called as, all of you, which is third one? Leucoplast. 3 types of plastids are present in a cell. So we know chromoplast is part of it. We know leucoplast is part of it. But then what is amyloplast? Amyloplast is a type of leucoplast. Leucoplast में 3 तरह के होते हैं. How many of you know? 3 तरह के होते हैं leucoplast में. Leucoplast is colorless plastid. It is colorless plastid. इसके अंदर 3 तरह के होते हैं. One is called as your amyloplast, which will come here. One is called as elioplast. One is called as ailuroplast. Okay. Here, amyloplast will store what? They can ask you a question. What is the reserved food material present in your amyloplast? What is the answer? Amylopectin. Amylopectin is a starch. Amylose and amylopectin is starch. So it mainly helps in starch storage. Now what is elioplast? Oil storage. Ailuroplast, protein storage. Aileron layer, protein storage. But tonoplast is basically a layer. Tonoplast is what? Tonoplast is a layer actually. Layer of what? Who can tell me? Tonoplast is a single layer present in which organelle? Who can tell me, student? Which organelle? किस organelle में हमारे पास tonoplast मिलता है? Anyone? 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 Tonoplast is present where? It's a layer around which organ? Who can tell me first? I'll see in the chat. Okay. Now, students. So tonoplast is not a plastid. It is not a yes. Vacuum. Very good, students. Very good. Very good. Now, this is present in the case of animal cell also. Is this present even in the case of animal cell? That is also a good question. That is also a good question, right? Yes. Vacuole, you will tell me. Vacuole plant cell very large. Answer I want. Next question. Next question on the board. Next question on the board. I'll give you students. I'll give you exactly. Let's say I'll give you 1 minute timer. Time starts now. I'll give you 1 minute time. Health is all about eating and fasting. You eat, you fast, balance is maintained. Okay. When you fast, when you are on vrat rakhte na vrat, hum nahi khate, khana nahi khate, for extended period, your cells clean out and recycle the intracellular garbage. The organelle responsible for this is, which organelle is called as suicidal bag? That is the answer. Which organelle is called as suicidal bag? Lysosomes. Easy question. Next question on the board. It's a very good question. Very, very good question on the board. Very good question on the board, students. Very good question on the board. A stain was developed by a group of scientists to stain a particular cell organelle. Stain is basically hamari color hai. It's a color. Pig, which can add color. Okay. Now, the stain was tested on various tissues derived from a ऑटोप्सी सैंपल ऑफ अ मैमल. एक मैमल के टिश्यू से हम बहुत चीज ले रहे हैं, निकाल रहे हैं. ठीक है. नाउ दीज ऑर्गेनल्स वर काउंटेड. द रिजल्टेंट शोड मैक्सिमम नंबर ऑफ ऑर्गेनल्स इन द सेल्स ऑफ द ब्रेन. सो इसमें से कोई ऐसा ऑर्गेनल है, देयर इज वन ऑर्गेनल इन ऑल ऑफ दिस व्हिच इज प्रेजेंट इन योर ब्रेन अ लॉट. ब्रेन के अंदर है, बहुत ब्रेन के अंदर है. ठीक है, ब्रेन के अंदर. ओके, ब्रेन. लेसर सेल्स इन हार्ट. हार्ट के अंदर थोड़ा कम है वो, थोड़ा least in mature sperm. Sperm के अंदर तो है ही नहीं वो. Okay, and absent in your erythrocytes. Erythrocytes red blood cells, उसके अंदर है ही नहीं वो वाला. Okay, identify the organelle present. Students, mitochondria it cannot be because mitochondria is present in your red blood cells. So it is present in your sperm. This is not the answer. Golgi cannot be the answer because Golgi helps in packaging. ER cannot be the answer. ER mainly does production of protein, but this organelle also does production of protein on the neuron. It is called as neurons protein factory. It is called as what? Neurons protein factory, called as Nissl bodies. Nissl bodies. So answer is what? Option number A is the right answer. Option number A is the right answer. Very good, students. Who answered? Who answered first? Is it Beaton or is it MIT? Is it Ayush? So this question has repeated. It is repeated in your last to last year question. Students, do you want to see the question? Do you want to see that question? It has repeated. Okay. This question has repeated before. After this exam. All of you, read this question. 2022 question. This is a question from 2022. 2022 question. Read this question. Nissl granules are in fact RNA bodies. In which of the following do they occur? Where do you find them? See, students, question is repeated. So this is the importance of doing PYQ. The importance of doing PYQ is that many concepts repeat unknowingly. So if you do all the PYQs from last 10 years, you have a very high chance that some question will repeat in the coming year, this year, and you will be like, thank you so much, sir. We attended your PYQ series, right? So very easy question. Yes, neurons. Easy, right, all of you? That's the question. Now, student, next question you see, student. Next question, all of you. Okay, look at this question. It's a very easy question. It's a very easy question. You can start. An organism has 27 pair of homologous chromosomes. How many chromosomes, students? How many chromosomes? 27 pair of chromosome. When I say 27 pair, of chromosomes, tell me the total number. Tell me the total number, students. One pair is 2. One pair is 2 chromosomes. One pair is 2 chromosomes. 27 pair is how much? एक पेयर का चप्पल, एक पेयर चप्पल, एक दो. Pair is always 2. Pair is always 2. 27 pair is how many chromosomes? First tell me, students. 27 pair is how much? 27 into 2. है ना? 27 into 2 we get a 27 pair. We tell in humans, generally in humans, we have 23 pair of chromosome. That is 46 chromosomes we have. We have 23 pair. Humans have 23 pair. But here they have given you organism which has 54 chromosomes in the body. It has 54 chromosomes in the body. Okay. Now, what is the question? In each daughter cell after completion of mitosis and each gamete after completion of meiosis 2, how many number of chromosomes you find respectively? Now, all of you tell me first of all, students, कभी भी माइटोसिस में क्रोमोसोम नंबर कम होता है क्या? कभी भी स्टूडेंट्स, किसी you take any mitosis, mitosis is called as reduction division. Mitosis is called as what? Sorry, equation. It is called as equational division. It is called as equational division because chromosome number never changes. So even after mitosis, it is going to be 54. Even after mitosis, it's going to be 54. Chromosome number never changes in mitosis. But does it change in meiosis? Does it change in meiosis? Yes. Chromosome number, students, remember, in mitosis, 2n will remain 2n. N will remain n. But in the case of meiosis, 2n will become n. Right? 6n is going to become 3n. 4n is going to become 2n. There is a reduction division happening in meiosis. So students, what is the answer now? Tell me. So mitosis will remain the same, but in meiosis, chromosome number will become half. Chromosome number will become half. So 54 chromosomes बन जाएंगे 27 chromosomes in after meiosis. समझ आ रहा है students? समझ आ रहा है ना question? समझ आ रहा है ना? So mitosis में हमेशा equational division, meiosis reduction division is happening. Okay? Easy question again. Now students, this is a question which is very good. We have learned this chapter. This is from plant kingdom again. This is from plant kingdom. See this. Given below are 3 statements about bryophytes. Bryophytes are lower plants. Yes, they are lower plants. Yes, they are lower plants, students. Okay. Okay, these are lower plants. These are lower plants with plant body differentiated into root, stem and leaf. Students, in bryophytes, do we actually find true root, true stem, true leaf? Never. Until your pteridophytes, listen to me very carefully. Until pteridophytes, you never find differentiation. Markantia dekha students? Have you seen Markantia? Students, have you seen Markantia in your life? Markantia. Dekha hai? This is a bryophyte. Markantia is a bryophyte. Here. Markantia is a bryophyte. Now, in this Markantia, do you see root, stem and leaf? In the Markantia, which is a bryophyte, do you see root, stem and leaf? We do not. This statement is wrong. Okay. Bryophytes are devoid of xylem and phloem. Yes. Xylem and phloem is present from where? Xylem and phloem will come into picture from pteridophytes. From pteridophytes onwards, we can see xylem and phloem. Pteridophytes से start होते हैं, है ना? Start होते हैं pteridophytes से. Now, bryophytes require water for completion of life cycle. Yes. Right statement, wrong statement. So what is the answer? Answer is 2 and 3. 2 and 3. Option number D is the right answer. Option number D is the right answer. Okay. So easy question it is. So you have to think a little. Now students, now. Next question on the board. It's a very good question again. Very good question on the board. Tejas, today I did Plant Kingdom today, 45 minutes. Okay. In some plants and fungi, some cell organelles are found which convert lipid into sugar. Students, please remember lipid into sugar. Always remember glycoxysomes. These are the organelles which convert lipid into sugar by glycosylate cycle. Glycosylate cycle, right? This is an easy question for all of you. It's direct PYQ hai to. Okay. Now, students. Students, I'm thinking should we do this question some other time, because this is multiple answer question. Okay, students, let me ask you all of you. Do you want a separate session for multiple answer question? Because here, if you notice this question, there are 3 answers are correct here. 3 are correct here. 3 are correct here. In this one, 3 are correct. So if we want, we will do a separate session of all the questions till now PYQs of multiple answer questions. Okay, students. So for this one, for right now, we will skip this question. I will include this question along with other chapters of your biology, and we will do a separate multiple answer question session. That will be killer now. Okay, that will be killer. Okay, because if you keep doing, otherwise you'll get confused. Okay, now students, now look at this question. Oh, okay, students, this question here is above NEET level question. Above NEET level. This question is about 12th level. About 12th level. This question is actually graduate level question. Like this is a graduate level question. We study this in BSc generally. Above 12th question or 12th grade. Students, why have you not liked the video? Students, all of you, please smash the like button. Mark your attendance, all of you. Okay, so this question I will teach you, students, because this is above NEET level question. Okay, generally we don't teach this also, right? I know many of you do not have idea. Many would have by-hearted this, right? Many of you would have by-hearted this, but do you want to see the real image of it? Do you want to see the cross section of equisetum? All of you, do you want to see a cross section of equisetum actually showing it? And this image can never be found in any of the internet. Whatever image I will show you right now is my personal collection equisetum section. Do you want to see my personal collection? See, all of you know I'm a botanist, right? So all of you know I'm a botanist, so I have multiple collection of multiple pteridophytes, multiple bryophytes, multiple algae cross sections I have. Okay, so I thought when I read this question, I realized oh, that's a good question. This is above NEET level. Okay, so for this question I have bought my personal collection of sectionings to show you some of them. Okay, now the answer is equisetum, students. Equisetum, it cannot be neatum. Neatum is a gymnosperm, right? So lycopodium also cannot be. It's a very exclusive to equisetum. This is the you can find this on internet. You can find this on internet. Here all of you can notice we have the valvular canals here, small large valvular canals everywhere. Canals can be found everywhere. Valvular canals are mainly responsible for the passage of air and everything. Then here, if you look at the vascular bundles right here, iske beech mein yahan par hota hai. Okay, there we have your cardinal canals here present right here. You can see here. This is second canal. This canal mainly helps in movement of water. Okay, but if you look at the cross section, students, these are my sections. Okay, copyright, I'll put a copyright on this. No one can copy this. This is my sections. Okay, this is equisetum. Yahan par thoda clarity nahi dikh raha, par can you notice here small gaps are present everywhere? Because this was taken in low power microscope. This was taken on low powered microscope, so it is not very clearly visible of equisetum. And equisetum is very difficult to take section also, because equisetum is very slimy. So difficult hota equisetum ka section lena. Okay, if I find better section, I'll show you sometime. But these are called as the canals here, canals present here. This is the equisetum section. Okay, now students, okay, this one is your Selaginella section. Here we can see steels, Selaginella sections. Okay, this one is xylotum. This one is xylotum section. It looks like a starfish, no? It looks like a starfish. This one is xylotum section. Okay, now students, do you want to see my favorite sectioning of all time? Do you want to see my favorite section which I have taken? I have personally taken, and it's my favorite section of all time in any plants, algae, bryophyte, pteridophyte, gymnosperms, anything. This one is my favorite one. This one is my favorite. This one is my favorite one. It is called as adiantum. Section, where it shows ectophobic solanostele. Okay, look, it looks like a heart, right? Looks like a heart, right? This is my favorite section which I have taken in my life. Okay, so I'm very proud of this section also. I always show this to my students also. It's my favorite sectioning. Okay, so students, if you want to see more of my sections, for example in cell cycle, I will show it to you. Like I have taken multiple stages of mitosis, multiple stages of your prophase one, right? Multiple stages of your metaphase. I'll show you all that in next class. Remind me, students. Remind me. Okay, remind me, students. Okay, remind me. I have taken multiple sections of your this thing also. What is that? Mitosis, immunology, students. Rest I can't show you because mostly it's botany, high level botany, right? Right. Now here I have certain questions. One more question. Let's see who can solve this question. This is from 2024. Last year question. This is last year question. Students, all of you quickly smash your like button and mark your attendance, all of you. See, this is a very good question and it's very easy. Many of you will not know what is lodicules and what is glooms. So let's skip this part. What you know right now. All of you will know, pine tree is a gymnosperm. Pine tree is a gymnosperm. इतना सबको पता है. Pine tree is a gymnosperm. And in gymnosperm, do all of you know, students, in gymnosperm, seeds without fruit. है ना? Seeds without fruit. So from this we can tell 4s3. So ये ऑप्शन चला गया, ये ऑप्शन चला गया, हाँ सॉरी, ये ऑप्शन चला गया, है ना? दो ऑप्शन चले गए अभी. ओके, 4s3. एक सेकंड, ये एक ऑप्शन चला गया. Then we also know, students, we also know banyan tree has something called as prop roots. So 2 is 4. Students, एक ही आंसर है अभी. 2 is 4 एक ही आंसर है ना? 2 is 4 एक ही आंसर है. पता है prop roots क्या है? Have you noticed banyan trees है ना, कुछ roots निकल के आते रहते हैं ऊपर से. कभी देखा है स्टूडेंट्स? कभी देखा है? एक banyan trees है ना, झूलते हैं उसके ऊपर, झूलते रहते हैं, झूलते रहते हैं, झूलते रहते हैं ना. That is called as prop roots. Prop roots from the banyan tree. Prop roots. Okay? Prop roots are present in banyan tree. There is something called as stilt roots also. Stilt roots are present in your maize and sugarcane. Stilt roots. Okay? What are stilt roots? Stilt roots is something like this. अगर ये maize है ना, maize, यहाँ से roots निकल के आते रहते हैं, roots. देखा कभी students? देखा है roots? From the lowermost node. From the node region, roots are coming out. In the case of maize and sugarcane, because stilt roots. स्टिल्ट रूट्स, ठीक है? स्टिल्ट नहीं देखा स्टूडेंट? देखना है अभी स्टिल्ट रूट देखना है कभी? अभी देखना है, दिखाता हूं, इंटरनेट दिखाता हूं, 1 मिनट स्टूडेंट्स. स्टूडेंट, हैव यू नोटिस दिस वन? एटलीस्ट दीस आर कॉल्ड एज स्टिल्ट रूट्स. ये देखो, कभी देखा होगा ना, इस तरह से निकल के आते रहते हैं. दीस आर कॉल्ड एज स्टिल्ट रूट्स. स्टिल्ट रूट्स देख रहे हैं यहां पर भी, देखो, ये देखो, शुगरकेन और मेज में दिखते हैं स्टिल्ट रूट्स. इसको बोलते हैं स्टिल्ट रूट्स. और जो banyan tree में होता है वो prop root होता है, prop roots. Then there is something called as pneumatophores also. How many of you know what is pneumatophores? Pneumatophores are your different types of roots which are breathing roots. These roots will come out of the soil for breathing. Okay, sir. Students, it's very helpful. PYQs are very helpful because you will realize what is the level of question you can ask. Okay, this is an easy question now. Now students, I will show you some neat PYQs. Let's see who can solve. Here is a neat PYQ for all of you. Neat PYQ for all of you, students. The organelles that are included in the endomembrane system. Endomembrane system. Which organelles are present in endomembrane system? All of you. Yes, mangrove. Mangrove has the pneumatophores. Negatively geotropic roots. Students, there's a small trick here. Every gamer loves victory. Every gamer loves victory. है ना? E for endoplasmic reticulum, G for Golgi complex, L for lysosomes, V for vacuoles. कहां पर स्टूडेंट्स? कहां पर सब कुछ? ऑप्शन नंबर डी. ये क्वेश्चन देखो स्टूडेंट, एक क्वेश्चन देखो अभी. Okay? Which of the following statements concerning the endoplasmic reticulum is incorrect? It is incorrect. It is incorrect statement. All of you, incorrect statement. SCR are sites for lipid. हाँ SCR lipid होता है, correct है ये तो, first one is correct, SCR lipid होता है. RER has ribosomes, yes, RER has ribosomes. SCR is devoid of ribosomes, हाँ. But this question, prokaryotes only RER? No. Prokaryotes do not have any type of ribosomes. Okay, students. Okay, prokaryotes do not have any ribosomes at all. See, prokaryotes में ribosomes नहीं होता है. Prokaryotes are primitive, very primitive. They are, students, primitive. Okay, this question you see, students. This question, see. This is last year NEET PYQ on the board. Last year NEET PYQ on the board. Okay, ये देखो देखो. The Golgi crystal ने are concentrically arranged near the nucleus with distinct convex cis or forming phase, है ना? So we have convex phase is also called as forming phase, है ना? Forming phase दिख रहा है. The concave or trans phase or maturing phase. So concave is called as trans phase or maturing phase. This is correct. The first statement is correct, students. Okay. Students, you have to check my lectures then. You have to check my lectures. At least then you will know all this. It's very easy. Okay. So, ये देखो student, दिखाता हूँ. This is Golgi. This is Golgi, right or no? This is Golgi phase. Here we have cis phase. Cis phase is here. Cis phase is here. Now, what is cis phase called as, students? Cis phase is also called as what? Cis phase is also called as convex phase or forming phase. है ना? Cis phase is also called as convex phase or forming phase. Forming phase. Now, समझो स्टूडेंट्स क्या हो रहा है यहां पर, समझो क्या हो रहा है. Now some molecule is entering inside. यहां पर एक molecule enter कर रहा है, यहां पर एक molecule enter कर रहा है, enter कर रहा है. Once it enters, the molecule enters, it undergoes packaging और बाहर निकल रहा है, यहां से बाहर निकल रहा है. So the main function of Golgi complex is packaging. Any type of your protein or lipid which is coming out of endoplasmic reticulum, किसी भी प्रकार का प्रोटीन और लिपिड जो एंडोप्लाज्मिक रेटिकुलम से निकल के आ रहा है ना, it undergoes packaging right now. It undergoes packaging right now. और packaging will always start from the cis phase, also called as convex phase or forming phase, और बाहर से निकल के आ रहा है trans phase, also called as maturing phase, also called maturing phase, है ना? निकल के आ रहा है स्टूडेंट्स, निकल के आ रहा है. Now this statement is right, but second statement, students, do you see the second statement here? The cis and trans phase of organelles are identical. Students, are they identical to each other? No, they are not. They are not identical. Both the phases look different from each other. Both phases are different from each other. They are different from each other. Okay? This statement is wrong statement. Wrong statement, students. Wrong statement. Okay. Next question on the board. Students, I've told you in one shot. Convex phase because of the cavity they make, because of the curvature they make. The curvature they make. Okay. If the pH of lysosomes is increased to alkaline condition, what will be the outcome? If pH becomes alkaline, will the enzymes function? नहीं, enzymes काम नहीं करेंगे तब. अगर pH बढ़ गया ना, enzymes काम नहीं करेंगे, है ना? तो hydrolytic enzymes will become inactive. All the enzymes will become what? Inactive. Because all the hydrolytic enzymes, they work in which pH, students? All the hydrolytic enzymes, they work in acidic pH. They work in acidic pH. अगर pH को खत्म कर दे तो, alkaline बना दे तो, all the enzymes will become inactive. Okay? Inactive, students. This is also very easy question. This is also a very easy question on the board. है ना? All of you know, cilia has something called as axoneme. एक cilia का structure देखा कभी? देखा है ना? Cilia and flagella. 9+3 combination. Students, all of you have seen cilia? All of you have seen cilia, yes or no? All of you seen the cross section of cilia? देखा कभी? देखा कभी students cilia का cross section? This is a cilia. Okay, this is called as a cilia. Okay, this is the cilia here. Cilia or flagella has the same structure. Now, if you look at the top view of this section, this is how it looks. This is the cross section of a cilia. This is a cross section of a cilia. Take a cross section from the top view. And at top views, we can see 9+3 arrangement of microtubules here. We can see 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 plus, right? 9 plus 2, right? Arrangement can be seen here. Now students, now. 9 plus 2 arrangement can be seen here. Okay. Now once you see this arrangement here, this is present in cilia. That middle part you saw there, right? 9 plus 2, so 9 plus 2. The middle part you see there, right? Middle part is called as what? Axoneme. Axoneme. Endoplasmic reticulum, ribosomes. Mitochondria, cristae. Kinetochore, spindle fibers. Kinetochore, spindle fibers. All of you know what is kinetochore? All of you know what is kinetochore, students? All of you know what is kinetochore? Yes or no? If this is a chromosome, right now, this is a chromosome right now. These are called as what? Sister chromatids. What is middle part? Centromere. Then around the centromere we can find the kinetochore. Around the centromere we can find the kinetochore here. Okay. Now on this kinetochore, all the spindle fibers will attach. Spindle fibers will attach on the kinetochore. That's why I said kinetochore spindle fiber, because around the kinetochore, the green color part, this green color is kinetochore. It's called as your kinetochore. Again, around the spindle fibers will attach. So if I make a question, spindle fiber attached to centromere, true or false? Spindle fiber attached to centromere, true or false? False. They will never attach to centromere. They will always attach, spindle fibers will always attach to kinetochore. Okay, I can change the question. This happens in which phase of your mitosis? Which phase of mitosis? Anyone? In which phase of mitosis? The spindle fiber will attach to kinetochore? Prophase, metaphase, anaphase, telophase. In the metaphase. In the metaphase, they go attach. This attachment happens. In metaphase, attachment happens. Okay. Now, students, very easy question on the board. Very easy question on the board. Very easy question on the board. Metaphase. Very good, students. If two chromatids, question is coming. Okay. Biosynthesis of ribosomal rRNA occurs in the nucleolus. Nucleolus is the factory of rRNA. It is a factory of rRNA. Factory of rRNA. Nucleolus is a factory of rRNA. Now, students, look at this question. Very good question on the board. Very good question on the board. All of you. Very good question on the board. See this. Which of the following stages of meiosis involves the division of centromere? Centromere gets separated in which particular meiosis? If you have attended my class on meiosis, you can tell this answer. If you have not attended in my class, you cannot tell this answer. Very simple as that. Now, let me explain you, students. Listen to me very carefully. In mitosis, does the chromosome separate? Listen to me, all of you. Listen to me. In mitosis, does the two chromosomes separate? Yes, they separate. They separate in anaphase. They separate in your anaphase. They separate out. But in meiosis, when do they separate? In meiosis, students, in meiosis, right? In meiosis, anaphase 1, what happens? In anaphase 1, homologous chromosomes separate. But in anaphase 2, what happens? The sister chromatids will separate. It will separate. So please be clear on what they're asking you. If they're asking you about homologous chromosomes, anaphase 1. If they're asking you sister chromatid separation, anaphase 2. So what is the answer right now? What is the answer? The question is the division of centromere. Division of centromere will happen only when the chromatids separate. Though chromatids separate ho jayenge na, chromatids separate honge. When the centromere splits, it will happen only in the anaphase 2. Anaphase 2. Clear, all of you? Clear? So they can change the question to something like this. During which stage of mitosis and meiosis respectively does the centromere split in anaphase of mitosis, anaphase 2 of meiosis. Anaphase 2 of meiosis they separate. Anaphase 2 of the meiosis they separate out. Okay. Or they can ask you a simple question like this. The dissolution of synaptonemal complex occurs during, pure concept question, dissolution of synaptonemal complex as well as the formation of chiasmata happens where? It happens in the students, it happens in the, I believe all of you don't remember this, right? Students, please remember, right? Leptotene, zygotine, pacatine, diplotene, diakinesis. I'll repeat after me, students. Repeat after me. Leptotene, zygotine, pacatine, diplotene, diakinesis. Let zebra play during day. Listen to me. These are the, see, prophase 1 has multiple parts. Okay. Let zebra play during day. What is L? L is leptotene. Z is zygotine. P is pacatine. D is diplotene. And D is diakinesis. Diakinesis. Okay? Now listen to me very carefully. I'll tell you in brief. I'll tell you. In leptotene, what happens? In leptotene, chromosomes become gradually visible under light microscope. In leptotene, chromosomes become gradually visible under light microscope. That's all, enough. What happens in zygotine, students? In zygotine, there is a bivalent and the tetrad formation. Now, what is the meaning of bivalent formation? Understand. In the case of your zygotine, what happens? In the case of zygotine, two chromosomes are coming together. Students, I mean to show you. This is easy to show my notes. It is very easy to show my notes once again, students. And just show you once. One second, students, give me 1 minute. Because this is very important conceptual thing which you have to know. Okay, see the students. This much all of you know, right? All of you know this much. Meiosis has interphase, then we have M phase. Meiosis 1, interkinase, meiosis 2. This much all of you will know. Students. Now, if you look at leptotene, what happens? In leptotene, chromosome gradually become visible under light microscope. That's all is enough. Now, what happens in zygotine? In the case of zygotine, two chromosomes are coming together. That is called as what? That is called as synapsis. In synapsis, two chromosomes are coming together. That is called as synapsis, and the formation of bivalent to tetrad. This is called as bivalent because of two chromosomes or two centromeres. It is called as tetrad because of 1, 2, 3, 4. 4 chromatids, tetrad stage. Now here, between the two chromosomes, synaptonemal complex formation is happening. Between two chromosomes, synaptonemal complex is happening in the zygotine. Now, in the pacatine, what happens? In the case of pacatine, there is something called as crossing over is happening. This chromatid will go this side, this will go this side, all of you. This chromatid will go this side, this will go this side. See here. Crossing over is happening. That is called as site of crossing over, where recombination nodule formation is happening. Recombinase enzyme is involved. Now, if you can notice here, after the entire thing, this chromatid came here, this came here. Now, this crossing over is happening where? Crossing over is happening in pacatine. What is crossing over? Exchange of genetic material between two homologous chromosomes is called as crossing over. Okay. Now, that is called as crossing over here. Now, the question which they are asking you is diplotin stage. What happens? Dissolution of synaptomal complex. जो बीच का complex था ना वो disappear हो गया पूरा, disappear हो गया. और उसके बाद there is an egg-shaped mark called as chiasmata formation here, chiasmata formation. Okay. उसके बाद diakinesis के अंदर termination of chiasmata and back to two different chromosomes here. So that is part of your prophase 1. Then there is metaphase 1. See what happens in metaphase 1, my notes. Metaphase 1. Then what happens in telophase 1? What happens in your entire meiosis 2, meiosis metaphase 2, anaphase 2. See, centromeres of cystochromids split up, anaphase 2, telophase 2. So these are my notes, students. These are my notes. Okay. So this is very difficult. You have to watch my one shot for that. This is a little high level concept, students. I can just show you mitosis also in small glimpse. I'll just show you a small glimpse of it, students. Small glimpse. See this. This much all of you little follow. All of you can follow. See here. This is your. This right here is your prophase. Metaphase. है ना? Metaphase. Telophase. So I given notes. Written notes for prophase 1. Written notes for metaphase. Kinetochore, centromere. Then written notes for your anaphase. What happens? Anaphase, centromere splits in mitosis. Then telophase, what happens? Okay, cytokinesis. In cytokinesis, plant cell, animal cell is different. Okay, so we will get there, students. We'll get there. I'll do one separate session, students. I'll do one separate session. Look at this question. Last question, see this. The oocytes of some vertebrates gets arrested for months or years. That happens in diplotin. Months or years it is arrested. Okay. So this is your homework question for all of you students. Students, watch my one Akar lecture, you will get to know. Just watch my one lecture, you do, students. Just watch my one lecture. Just type mitosis and my name in YouTube. Just type mitosis and my name. Watch that 1 hour lecture. It's just 1 hour lecture. 1 hour lecture, and every concept of mitosis will be clear to you. Every concept of meiosis is clear to you. 1 hour lecture it is. That's all. Just check that once. Okay. This is your homework question in comments. And all the students, next class on PYQs. Do you want on genetics? Do you want on ecology? Do you want on plant physiology? Do you want on human physiology? Let me know in the comment section which one do you want in the next class. Okay. Okay, students. So let me know in the next class. Let me know next class which one do you want. Next PYQ from my side, because when you do a PYQ, you revise a lot, you learn a lot, and you make mistakes and you grow. Okay. So all the best, students. Thank you so much. I will see you in the next class. Until then, keep studying, keep working hard. Bye bye all of you. Students, again, if you have not filled, students, if you have not filled the revision camp, please go check the description of this video. Check the description of this video. You will notice there is a link. Click on that link and students, you will have access to the revision camp. So fill the link, add the details and be the part of revision camp as soon as possible. Because imagine, abhi is tarah se hai. Revision camp mein kya hoga? Revision camp will be double the fun. Okay. So students, thank you so much, students. Comment section, answer in the comment section. Okay. So bye bye all of you students. Bye bye. Bye bye students.